Thank you. Thank you for the moderator for the introduction. So my name is Yan Liu. I'm a radiologist by training and Chief Medical Officer of Media and Technologies. Today I'm very happy to present to you iBiopsy in lung cancer screening together with my colleague, Dr. and Professor Benoit Yue. And so maybe couple of words about Median, what we are doing. So Median, we have our headquarters in the south of France, and we operate also globally. We have office in China, in US, and also we provide imaging services to our pharma clients as well. But we also have the innovation in our DNA. This is why that we continue to invest in the R&D, and that's the iBiopsy we'll present to you today. And iBiopsy is a technology in AI, in a medical software, and we will use that for our patient care. And today we will focus on lung cancer screening. So iBiopsy, we're trying to also shifting the cancer diagnosis paradigm. And we are focusing also today in liver cancer and lung cancer. So why we care about lung cancer? We see that lung cancer is still the #1 cancer killer that, based on the 2020 data, that contributes to 18% of the deaths of the entire cancer. That results in 1.8 million of deaths. And also we see that the survival is significantly related to when we find the cancer. For example, if we find it early, for example, in the stage 1, the 5-year survival is above 68%, even can reach to 92%. However, when we find them in the stage 4, then the 5-year survivalship is only less than 10%. And unfortunately, that presents more than 40% of the population. So we really need to have a cancer screening program to detect cancer early. And now in US, cancer screening is also part of routine. But however, looking to the data, 2021, the compliance of lung cancer screening, there was only 6%. That represents only 870,000 patients. So now it's already in the reimbursement. It has been in the guideline. Why patient or why high-risk population are not following that? There were several reasons. First of all, people feel that, I already smoke, I don't want to have a bad news. Or there was also this kind of low awareness. People don't know that. And also, very frequently, if we have a nodule, patient will also have anxiety. Is that a cancer? So those kind of things could impact the compliance. And also there was some feelings, okay, lung cancer screening, there was a very few percentage of patients will find, less than 5%. Maybe I will not be the bad guy. So people feel that the incidence is very low, so not following the guideline for the lung cancer screening. And also the last is, even if that we have all the patients go for lung cancer screening, do we have so many radiologists to read those reports? I think this is also something in Europe people think a lot, is if we have those people go for lung cancer screening, we have enough radiologists to find those nodules, to detect and to report, etc. So therefore, iBiopsy, so our system, is trying to provide a solution. So iBiopsy is aiming to identify the malignant nodules and also reduce the false positive, and in this regard also decrease the independent nodules. So why we believe that is important and what's the differentiators that we're offering? So we offer a system which is end-to-end. First, we will detect the nodules. So in this regard, if radiologists, due to the high workload, may miss something, that we can find it. And then when we detect it, the system will also characterize it. So is that a cancer or is it a benign nodule? So in the way that we offer an entire solution for the nodule detection and characterization. And in this regard, we believe that could help our radiologists to have better diagnosis, find a cancer early and accurately. So iBiopsy is a software in lung cancer screening, and it is also AI, machine learning technology, end-to-end CAD-E and CAD-X, as I mentioned before, detection and characterization. And this will be allowed to have early detection and characterization of lung nodules, in this regard to better manage patients in the lung cancer screening framework. And it's also intended to add the identification of cancer to provide a patient a better care and avoid unnecessary procedures. In this regard, also saving the cost. Now, I'm very happy that my colleague Benoit to give you our recent data. And Benoit, please.
Thank you, Jan. Thank you, Jan. So let me take you through the workflow that we are going to provide for both lung and liver software as medical device. For today, we're presenting results on lung, but we are also working on liver diseases and HEC in particular. So our platform is going to be available either as a service on the cloud or on-premise, and will work in 4 very simple steps. So the patient will be scanned, as usual, in CT, in the case of lung cancer screening. The DICOM will go to the PACS, again, as usual. We are going to provide a gateway that is going to receive the DICOM that needs to be analyzed by iBiopsy LCS. And the gateway is going to send to our servers, either on the cloud or on-premise, for analysis of the DICOM. I'm going to detail the algorithms that are within the analysis in the next few steps, but for now, we stay at this level. The results are going to be computed and organized in a DICOM that is going to be fed back onto the PACS, and when the radiologist will be analyzing this patient, both the report and the original scan will be available for analysis. This will allow quicker and also maybe more precise diagnosis, and this is what we will show through our FDA validation in the next few months. So this is the overall workflow. Let me dive into that green box you see at the bottom. Or, I'm going the wrong way down. Our state-of-the-art artificial intelligence and data science detection and segmentation for lung cancer screening in the framework of this CADI-CADEX. So again, we're receiving a patient low-dose CT scan, and this CT scan is going to be analyzed, first of all, to identify the location of the lung. This is the organ we are looking for, and this is what we concentrate on for this product. Within the lung, we'll be looking for nodules and findings. Those nodules are going to be segmented. Each of these nodules is going to be characterized in order to identify whether it is a benign or whether there is a certain level of malignancy. We're also going to compute other descriptors, such as volume, mean diameter, and all these are going to be fed into the report that can be used by the radiologist or any other clinician that actually needs this type of information. And this is the report that will be available as a DICOM on the review workstation alongside the original scan for the clinician. So let me go into each of the steps that were visible on the previous slide and show you the level of performance we are able to achieve for each individual step. So the first thing we need to do is identify where the lung is. Okay, so for this, we developed some AI that has been trained on the public dataset known as LIDC. We got this dataset re-annotated by our expert radiologists, and this allowed us to train some deep networks that an ensemble of deep networks is now providing us with information about which information actually is lung and which is not. We are providing a sample of our performance based on 80 scans from this dataset. And as you can see, the dice, so the dice is giving us information about how close we are to the original segmentation provided by the radiologists themselves. If we had 100%, then it means we are exactly equal. If we had 0, it means there is no overlap between what we specify as being lung versus what the radiologists think is lung. So you can see we're pretty close to 100. Therefore, our algorithm can be trusted on that particular step. So this is the first step. We know where the lung is. We can continue, dive into the lung looking for nodules. So for locating, detecting nodules within the lung, we continued using this LIDC dataset and focused on a particular challenge that was actually addressing this issue, the Luna 16 Grand Challenge. It's providing us with 888 patients that have been annotated by 4 experienced radiologists and providing us with a large number of nodules that can be used for training, once again, yet another deep learning architecture. As you can see, for the purpose of comparison with the literature, we are able to achieve with this architecture almost 95% sensitivity for 1 false positive per scan. Of course, this is not the model that goes into the product. We have actually trained with a lot more data coming from an LST, for example, but the same architecture is being used as a stem for the entire algorithm. As I mentioned in the original slide, when we have detected nodules, we want to segment them in order to be able to compute additional information, both for characterization, but also for providing details about their size, and this is an important criteria for malignancy. So for this, we continued using the LIDC train ensemble of deep networks, and you can see some of the results that we are able to achieve on that image on the right. When we have done the segmentation, this allows us to very easily compute the volume of this particular nodule as well as the mean diameter. So we're moving along and we reach, I guess, what most of you have been waiting for, our characterization performance. That means our ability to predict, looking at a specific nodule, the level of malignancy. So in this case, we have used still LIDC, partly, but most of our training data comes from NLST. Almost 10,000 patients have been fully annotated by our team of radiologists, and this allows us to train high-quality models that are going to be used by the iBiopsy LCS product. So in terms of test, our test is focusing on NLST. Over 2,000 patients are being used, completely independent of the training sets. As mentioned on the slide, this results in a quite large number of nodules to analyze, over 37,000 nodules to be analyzed in terms of malignancy. So this time we are using a number of approaches to characterize each of the nodules. We are using traditional machine learning on radiomics. We are using a new morphological feature that I'm going to show in the next slide, as well as combination of deep learning models, both in 2D and 3D. And actually, both this combination of model and the 3D morphological approach that I'm going to show are actually patents that have been filed and hopefully will be accepted within the next months. Enough about how we do it. What kind of performance can we expect from this architecture? Well, in terms of AUC, as you can see on the receiver operating characteristic test in that diagram, we have performance of 97.4. This is an improvement over our previous models, thanks to the addition of all these different features, deep neural networks, 3D morphological features, as well as radiomics. In terms of usability for the radiologists, well, sensitivity is always very important alongside specificity. We don't need to compromise. We can have both with very, very high performance. As you can see, the max unit index, that's a point characterized by the little cross you can see on the top left of the curve. We have 96.5 sensitivity for 97.2 specificity. So this allows to no trade-off between malignancy and benign. So as I was mentioning, we are combining different approaches to achieve this very high level of performance. And one of the things that we've been working on at Median is new feature descriptors. And here I'm showing one of the feature descriptors we've been working on, this morphological feature that allows us, from the segmentation of each of the nodule, to provide a way to characterize this nodule in terms of its local curvature. So you have here two nodules, one very spherical, and this happens to be a benign nodule, and you see, this doesn't work, right on the left of that nodule, you can see an histogram, and this histogram is the representation of the curvature of that nodule. On the right of that nodule, you can see a malignant nodule, very speculated. And you can see the histogram of curvature that this nodule, that describes this nodule. And you can see it looks extremely different from the previous one. Okay? The response in terms of local curvature is completely different. And if you look at a large collection of nodules from an LST, for example, you can see that, and this is what the bottom right figure shows, you can see that the average distribution in terms of mean curvature is very different for benign nodule than from malignant nodule. So malignancy in lung nodule is not just about size. Okay, and if you look at the papers that are mentioned on that slide, the ESMO 2022 and Mikai paper, on both these papers we are showing the importance of this additional feature representation, as well as our deep network features, to characterize and to diagnose, in some sense, each of the nodules that we can detect. If you want more information, of course, we are here at booth AI35, if I'm not mistaken. So please come to talk to us and discuss. What we've been presenting here today is one part of the work we are looking towards oncology, our lung cancer screening product, which is AI-based, and is going for FDA validation in the next few months, and we expect clearance in the first half of 2024. Our iBiopsy LCS benefits from end-to-end imaging workflow support, that is from the DICOM back to a DICOM right in the radiologist's hands. Every pulmonary nodule is identified by the system, so this allows us to very quickly provide feedback to the radiologist and potentially improve his workflow. Of course, we want to, thanks to this product, we want to minimize the number of false positive, but also false negative, and reduce the number of unnecessary biopsies, so interventional procedures. Again, here we are only presenting our lung cancer screening product. We are working in parallel on an incidental pulmonary nodule product, basically driven from this original one. Liver cancer for HCC detection of small detection and characterization of small nodule that's below 2 centimeters, and we're starting to have excellent results on this. And on liver, we're also working on NASH and diagnosing fibrosis and scoring diagnosis. With this, I hand the talk, and if there are any questions, we'll be happy to answer them. Thank you.
Are there any questions from the audience? No question. I was wondering on the... Oh, there's a question there. I wonder if...
Hello, sir. Thank you for the good explanation. I just wanted to ask about the ensembled networks, because it was at page 9, you were just saying there was the network is assembled, and I just wanted to ask if it's okay to tell us how it was assembled. for
Oh, sure.
2D and 3D networks?
Okay, so what we're doing is instead of using a single network, we are training multiple networks, forcing them to learn different things, a bit like you have different radiologists looking at a single scan, and potentially you want to agglomerate all the information from all of those experts to make a better informed decision. So maybe 3 of the N and deep networks are going to go for malignancy level that are low, others are going to go for very high, and then you need to learn how to combine these. When can you trust an expert? When should you not trust an expert? And this is part of what we do within this product.
So the ensemble was about 2D and 3D networks only, just of these 2 networks. And
So that's later on the slide. Yep.
it is hard to explain about the details right here.
All right, so, well, unfortunately the patent is not yet available and we haven't published much on this, unlike on morphological features that actually has been reported in 2 papers. What we've seen is that there are things that are better trained or better learned in 2D rather than 3D. So instead of just doing 3D things, we benefit from having multiple experts looking at individual things and learning individual things, and then combining all of this together, to put it short.
Thank you for the explanation. Thank you.
Any other question? I was also wondering about the variability of radiologists that segment nodules for the training. How high is that variability? And if it's high, how did you then take it into account to identify the ground truths?
Yeah.
How do we mitigate?
Yeah, maybe I can take this question because we have also very close discussions with the FDA. They do require have 3 truth ring, 3 radiologists. So our current sample is so we included 1,000 patients, right, for the segmentation piece. Yeah, but we are doing more now. The thing is, when we have 3 truth ring, we will also have the variability of each readers, and this is still under the analysis. So we'll be happy to share with you very soon. Yeah.
Thank you very much. If there's no further questions, then we can end the session here. Thank you very much again. Impressive results.
Thank you.